Great. Well, I want to thank everyone for joining us here at Cota Access. I'm Janis Naeve. I'm partner here at Cota Capital, and we're very fortunate to have today Dr. Robert Loberg. So Robert Loberg is the Vice President and Lifecycle Leader on Oncology and Genetics at Roche, where he oversees development and commercialization for the next generation sequencing and the PCR portfolio, the CLIA lab, and the pharma partnering and services business. Robert has held roles across R&D and commercial activities at both biopharma and diagnostic companies. He's generated strategies that has produced a broad portfolio of pharmaceutical and diagnostics products. He has over 50 peer-reviewed publications and has done considerable hands-on lab work. And he's coming from a demonstrated passion for the biomarker research and its application in cancer therapeutics. Robert received his Ph.D. in physiology from the University of Michigan, where he also served as a research assistant professor, Department of Internal Medicine and Urology. So let's start by learning more about you, Robert, and what has brought you to where you are today.
Thanks, Janice, and thanks for that, very warm introduction and welcome. I appreciate the opportunity to be with you today. It's an interesting story in terms of how I got to where I am today, because if you would have asked me 20, 25 years ago as a young kid graduating undergraduate, I never would have probably identified this as the place where I would have ended up. But, life's journey is almost never in a linear path. It's always jagged. So really what happened for me was, as I graduated undergraduate from Baylor University, I was very interested in medical science. Of course, like most kids, was interested in becoming a medical doctor. But I got involved in some research at the time and really kind of caught the bug for research. So ended up joining the University of Michigan in a Ph.D. program where I grew up doing, research in clinical departments with some investigators who were running a lot of clinical trials, right, at the University of Michigan. And one investigator in particular, a gentleman named Ken Pienta, who was my postdoc advisor and mentor, got me involved after I graduated from the PhD program and got into my postdoc with him. And he had just started a rapid autopsy program for prostate cancer, where he was consenting patients who lived within a certain close radius to the hospital that upon the time of their death, they would come into the hospital and allow us to actually do a full autopsy and collect tissue for research purposes. And I ended up, as part of my postdoc, running that autopsy program, and really at that point observed really the devastation that cancer has on the human body, and not just the devastation, but the wide variety of presentations that cancer has when you're performing something like an autopsy and you actually see the disease as it metastasizes in all different ways in different people in a single disease that we call prostate cancer. And so part of the research that we got into at the time was really trying to understand why prostate cancer looked a certain way in some individuals and a different way in other individuals. And that was really, for me, the launch into understanding what we now call personalized medicine, or at least patient stratification, which leads to personalized medicine. I then went on from there to join a pharmaceutical company called Amgen, where I had the fortune of being in the group and leading the biomarker and diagnostics function for about 13 years, and was able to get involved with and see a lot of applications of how understanding disease biology at the level of an individual patient can really help inform and drive the way we develop new and innovative medicines, but also think about treating patients differently that's much more tailored to the individual as opposed to a broad group of people that we would classify as having the same type of cancer, like a prostate cancer. And from there, that's where I really caught the passion for bringing together the biology, the medicine, and the tools used to measure that biology and driving patient care, which then led me to Roche Diagnostics, where now I'm overseeing a portfolio of products that we build and applications around developing these tools to measure these biomarkers to drive personalized medicine, personalized cancer therapy.
Excellent. It's interesting. I always learn things, even though Robert and I have known each other for well over 10 years from Amgen. I did not know about your background that way. I mean, I did my graduate work at USC and did a few trips to the pathology lab over there at County General. So
Yeah.
it definitely brings it together when you're working with pathologists and you're seeing tissues and correlating with disease. It definitely does bring it together. So, as most of us know, cancer isn't a single disease. It's really heterogeneous and complex. However, it has been described as a disease of the genome. So fortunately, with advances in the DNA sequencing, we have amassed a great deal of molecular information that has advanced cancer research and treatment in a direction that helps us personalize cancer. So sequencing the genome is what we would say necessary, but not sufficient. What else was needed to actually get us to where we are today with therapeutics and diagnostics?
Yeah, it's to no surprise that being able to sequence the cancer genome was such a huge step in the industry, the healthcare industry, for understanding biology, patients, and therapy in a very different way. But of course, it didn't start with cancer genome sequencing. We were starting to consider how to use biomarkers, whether they were protein biomarkers present on tumor tissues through technologies like immunohistochemistry early on, or even blood samples where you can measure circulating proteins to help understand the differences between patients. But I would say clearly there was a quite significant inflection point when the industry became successful in sequencing the cancer genome, and we started to really get a window into individual cancers and how unique they are in a way where we used to clump cancers together and think of them as one group of diseases of one group of diseases. I think just sequencing the genome was the first step, though, the cancer genome. What's really happened since that first ability to sequence the cancer genome, which took $1 billion and 10 years to do, fast forward, we're now at a place where we can sequence a single genome in about an hour on some of the newer platforms and chemistries that exist out there. So the speed for which the technology that enables this type of work to be done has just exploded over the last 10 to 15 years in a way that really has changed our ability to generate the data. In addition to that, I think the other things that have happened around being able to generate the data is the research, both the basic research and the clinical research, in terms of how you actually use that data to interpret disease biology or a patient's response to a given therapy, or even how we think about innovative ways to design different types of drugs in ways that we couldn't before we had this information. And so it's really coming, I think, all together into this kind of current state we're in, where we have the technology, and the technology continues to advance, and we can talk some more about that, later as well. The technology continues to advance that allows us to do things that we haven't been able to do before, or do things in a way that is faster, more cost-effective, easier, right? Some of those categories, but also our understanding of how to investigate, interpret the data that comes off of these new technologies and apply them to biological or clinical questions is really what's making such a big difference in our ability to really advance this whole concept of personalized cancer therapy.
No, it's very interesting. And obviously, Cota's quite interested in the health tech space and, next gen sequencing. But then there's also a lot of other technology and advances and single cell sequencing that's coming on board, and different life science tools, different ways to do the preps, et cetera, that there's a real culmination of the technology, which is truly empowering, right? And really brings it down to a more personalized level. So, certainly we've all heard the terms, personalized cancer therapy, right? People kind of understand, what is that? What exactly does that mean? Right. And certainly from our backgrounds, maybe we have a little bit deeper knowledge of what generated a targeted therapy and what's now personalized therapy. Could you you help everyone who's listening kind of understand, how do you frame that personalized medicine or personalized cancer therapy?
Yeah, I mean, it's a great question. And to your point, there are several different ways that you can think about personalizing therapy for a cancer patient. Everything from identifying a subgroup of, let's say, non-small cell lung cancer patient tumors that have similar genomic mutations or characteristics for which you could develop a medicine that specifically goes after or targets that subset of lung cancer tumors that carry those similar characteristics, and develop a targeted therapy that targets a specific subtype of lung cancer, but is applicable to any patient who has that subtype of lung cancer, all the way to understanding what is unique about your lung cancer versus my lung cancer, and actually creating an individualized medicine that targets just your lung cancer versus just my lung cancer. And we certainly use a lot of the sequencing capabilities to be able to do that. We also use other tools that give us insights into the differences between tumors and the tumor biology associated with it. But that's not even where it stops, right? That's really, for me, where it starts, because then going beyond that, now within the industry, we're thinking about technologies and tools that not only allow us to understand the biological differences between two cancers that come from the same tissue of origin, like lung cancer. We're now thinking about technologies that we can use to identify that either really early, before somebody has clinical manifestation of the disease, the physical symptoms that takes them to the doctor to then be formally diagnosed. And there we get into these concepts of early cancer screening and whatnot that allow us to identify those who are at high risk of having an undetected disease, for which we could then conceive of ways to intervene and run clinical trials and develop medicines that may prevent these early cancers from becoming really significant disease burden tumors that arise as metastatic disease and have really horrible outcomes. But also on the other end of the spectrum, when we develop these new medicines and we treat patients that have a particular tumor type, we can now in a blood test start following the level of tumor that's detectable in a blood sample, and we can monitor the effectiveness of these drugs. We can monitor it to the point where it's no longer detectable in a blood sample, and then we can monitor the patient to see if the disease recurs, relapses, if that patient relapses really early with a detection in a blood sample. So we're using these technologies not only to understand the disease biology, but really understand the patient journey and how we can actually tailor treatment patterns to an individual patient's journey, not just a targeted therapy or an individualized therapy that's created based on somebody's, tumor, what the tumor expresses or is made up of.
Yeah, I think what you just did is you probably leapt through a few decades of research and drug discovery development and the diagnostic component as well. Because I think back to like targeted therapy, like when I was in later graduate school or postdoc, Herceptin, right, for breast cancer. When that was really kind of, then you diagnose a breast cancer and then you could figure out what to treat it with, because there's been some discoveries around those lines. But then as you started to expand this, it's like we can dig down to a type of cancer, a single cell within those cancers. And you talk a bit about kind of even looking at it longitudinally over time, right? Within that even the patient journey, right? It's a patient journey from the tumor, but it's also even a patient journey from the time of diagnosis to determining a treatment, to actually then monitoring it, to help with the rest of the treatment cycle, right?
Absolutely.
Can you maybe spend a little bit more time there going into, because that's really where the therapeutic and the diagnostic starts to come together, and talk a bit about what the impact of that is, right? Where if you can diagnose early, what that means. If you can find the right treatment specific, what does that mean? And once you treat, how is it that you can track it to make sure your treatment's working or something like that? So just spend a little bit more time, on that area, if you will. And an area that we're quite interested in is minimal residual disease. Maybe, kind of give an example of how you do that.
Sure, absolutely. It's a large question, so we'll see if we can break it down into some parts. So when I think about our ability to really generate this kind of information and understand disease biology, what I really like to think about is, over the last 10 years since the Herceptin days, when a drug and a diagnostic test are paired together in a label that guides a physician to treat a specific type of cancer that has, for instance, a genetic mutation, it's called a companion diagnostic, for those who aren't familiar with that word. The first companion diagnostic was HER2 as it related to this drug Herceptin. That was 15 something years ago. Now there are 45 or 46 approved biomarkers as companion diagnostics that relate to about 85 to 90 different targeted therapies that are currently approved through the US FDA.
Hey, Robert, can I just ask you a question? Can you tell people what a biomarker is? I mean, we're
sure.
interested in those words. Describe what a biomarker means.
Sure, absolutely. So, in our industry, when we talk about this concept of a biomarker, it's really something biological that we can measure with some tool technology, and we can assess whether or not it's there, or if we can actually measure how much of it is there or not. And when I say that, I think most people, if you think back to kind of your high school or college biology classes, everybody knows DNA, I think, and RNA and protein. Well, all of those are examples of biomarkers that we can measure with different tools. And so Herceptin was a protein that is expressed on the outside of tumor cells, right, of breast cancer, that we can measure with a technology called immunohistochemistry. It's a slide that a pathologist will stain HER2 for HER2 with, and then look under a microscope and actually look to see if HER2 is positive, it's present or not. And then they can quantify how much of it is there, and that tells them something about the tumor itself, where it is in its evolution, if you will. And then ultimately what was proven in clinical trials is that you can use that information to say patients that have high levels of presence of that HER2 protein biomarker, if they're treated with a particular drug that targets that HER2 protein, those patients do really, really well in terms of an outcome or a response to that therapy. And so when you think about DNA and sequencing, if we can go back to that, every cell in the body has the same DNA in it, except for cancer cells. When the genesis of cancer is that the DNA starts to change for a variety of different reasons, we call it mutating. When the DNA mutates into something that is abnormal or non-self, and we can detect those abnormal changes in the DNA of cancer cells through these sequencing technologies. Originally, that started with being able to take a biopsy from a tumor, and taking out the DNA of that biopsy and putting it on these sequencing machines and being able to sequence and look at and compare the tumor DNA to a normal sample of DNA and look for the differences. That's how we do that. Nowadays, the technology has really advanced to the extent where you can do that out of a 8 milliliter regular blood draw that you would get at your CRO when you go in to do your routine clinical lab blood draws for cholesterol or whatever else. So now we're getting to the point where we can actually do this in a much less invasive way. We don't need surgery. We don't need these awful biopsies where you have to go in and extract tissue. You can do it out of a blood sample, or even potentially out of something like a urine sample, or other ways to collect the material. At the end of the day, all of the technologies and all of the purpose of the technologies is to get to the same question. Can we detect the presence of tumor DNA, and can we define what abnormalities the tumor DNA has in that individual, which allows us to understand how that individual is the same or different than everybody else who has that same cancer type. And then go on to ask the next question, which is, can we treat those patients differently with specific therapies that target that mutation or some other way that improves the response and the outcome for that patient versus another patient who doesn't carry that same abnormality seen in the DNA. So that's how we use kind of biomarkers, and really it's a biological sample that we can measure something in. That's what we call a biomarker. And we're using those. So going back to kind of the approvals through the US FDA and around the world, there's been this increased understanding of the underlying genomics of tumors that allows us to really take something like a lung cancer, and we just don't call it non-small cell lung cancer anymore. If you really look at non-small cell lung cancer, it's probably 12 to 13 different subtypes based on common genomic alterations or abnormalities, differences than others. And companies have developed very targeted therapies that go after certain parts of non-small cell lung cancer patients based off of that information. And it's really allowed us to advance the way we think about, again, treating, understanding, and thus then treating patients with a similar disease. But for me, I mean, yes, that is absolutely exciting, and we continue to explore ways to do that. But the real exciting thing for me is then how do we use that information or build additional diagnostic technologies to allow us to look at things like you mentioned, minimal residual disease. So that's going into a blood sample with a very specific test to measure the presence or absence of tumor in a blood sample to such a sensitivity where we can say that below the ability to detect that tumor DNA in that blood sample, we can no longer detect it. Therefore, there is no more disease, right? Detectable disease. And when you look at something like multiple myeloma as a disease where this concept of minimal residual disease is probably most advanced, what's happening is we're understanding that some of these novel therapies that are coming out are able to drive down the detectability of tumor DNA in a blood sample to a level based on these technologies, because we can get really, really sensitive in our ability to detect the presence or absence, that patients who achieve an MRD negative status will go on with a very significant prolonged period of time where they no longer have the clinical manifestations of the disease. They can come off therapy, they return to, let's call it a normal life, but we can continue to monitor with a test so that we catch, if that disease does come back, we catch it really early before it becomes spread all over the body and causes a lot of the clinical problems that patients experience, which of course then ultimately lead to really bad outcomes. And so this ability to develop technologies and understand how to use them, not just to profile tumors and create therapies, but to understand the patient going through their journey of diagnosis, treatment, response to that treatment, and hopefully then a period of time where they don't have any presence of disease, and then look for signs of early relapse of disease, really creates a very new way for doctors to really think about their patient care and their patient management.
Yeah, it really does, I think, come down to that, the patient and the physicians, and giving them, the physicians certainly, the tools to monitor their patients better. And the patients, you know, in some senses, kind of a peace of mind to know that it's being tracked, right? And not waiting to see some other signs. Yeah. So, and, you know, you and I are both, you know, a bit, you know, technology technophiles, right? And this is not a trivial task, developing these technologies, right? And, you know, obviously Cota Capital, you know, we're investing in health tech. So there's a number of companies that are developing new platforms and technologies to do these things, right? Yeah. So it takes more than a village. It takes an industry to do this.
Yes.
So that's one thing I would like to kind of hear your scope, because, you know, while you are at Roche and you have certain responsibilities and tasks, you're also, you know, have this radar out to what else is going on. And then even within those contexts, layering on, because I'm sure a lot of people who are listening in have heard the term omics, right? So what does omics mean, right? You have the genomics, but build on it. And where did those technologies come from to detect that? And how are you seeing that information being deployed to improve this whole kind of process?
Yeah, no, you're absolutely right. It's a great question. When I think about the omics, you can put omics on just about the end of anything right now, right? We have genomics, we have proteomics, we have metabolomics, we have all kinds of omics. Really, for me, what omics means is you're just looking across lots of different variables, right? Whether it's DNA and protein, DNA, protein, and bacteria in your stomach, right? This whole concept of microbiomics, which is another one. What we're trying to do is understand disease in a different way based on the biology, right? So that's kind of the first level for me. The next level is the tools and technologies we need to be able to identify, measure, and or quantify those different omics, if you will, and generate data that we need to analyze in a different way. And that's where, when you think about sequencing is much more than just the DNA that goes onto a sequencer and the sequencer machine reads each base pair and spits you out a sequence at the end. It's all of the tools needed to collect and prepare the sample up front to be able to go onto the sequencer. It's the age-old adage, garbage in, garbage out, right? So the upfront stuff has to be really, really good to go in and get really good quality results coming off of a sequencer. The other end of that spectrum is the bioinformatics needed to take that information off the sequencer and do the types of analysis that we need done to be able to have quality interpretable results coming off the sequencer.
Yeah.
But it doesn't stop there, right? Then you have to take that information and put it in context of a patient, of biology, of disease or whatever. And that's really that whole kind of what we say end to end, sample into, data out and contextualization of it really is really key to being able to do this successfully. And then, underneath that is doing the type of clinical research and clinical trials needed to say, yes, we can measure these different omics, these proteins, genes, whatever. We have the tools to do it, and we can collect the data and analyze the data in certain ways. Now we need to test the hypotheses, right, in the context of these clinical trials to know if any of it matters, right? Or if it's just noise.
Right. No, I mean, you hit on a lot of key things that, we could bring you back multiple times to dig into that. But what I did key on is the amount, when you said the bioinformatics piece. And I think that that's where there's a huge explosion because, so much data comes off of this analysis, right? And, I came across a stat that said 30% of the world's data is healthcare data. Now, that's more than even just what here. It's like a lot of the other social terms of health. So there's so much data. So this is really kind of a shout out to like data scientists training and bioinformatics is that, generation of data, the analytics, the right questions to ask for the analytics and the iteration to go with the insights to come back to develop better diagnostics and better therapeutics. So, but more on that later. That's, that's too much to cover on right now. But I did want to close by thanking you, but I also want to do a quick rapid fire question for you.
All right.
Okay. What was your favorite book that you've read recently?
The Long Walk. It's a great story about 1941, a prisoner of a labor camp in Soviet Union and escaping and making a trek down to India, to the British India province down there for safety. Story of perseverance, humanity, and just a really great read.
Excellent. I'll write that one down. What do you have a favorite food that you used to hate and now you love?
Brussels sprouts.
Same here. You throw some bacon on it, it's all good.
Bacon, that's it.
Bacon, dress it up with bacon. And this one I really am interested to hear. It's like, if you'd not gone down the path of being a scientist, what would you be doing? And don't worry about skill set. skill set aside, what would you be doing?
Oh, I would say professional golf. I started and grew up playing golf all through high school and loved it, and then decided to take a real job where I had friends who stuck with it and got to travel the world and play phenomenal golf courses. So I haven't decided if I've made the right or wrong decision yet.
That's okay. At least you can enjoy it as a side sport for sure.
There you go. That's right.
Excellent. Excellent. Well, so we'll continue to chat a little bit. I need to go back and show the our legal disclaimer slide so we can get through that. Let's see. Oh, there we go. So Robert, it's really good to see and hear you again. Like I said, we'd interacted for, a number of years at Amgen and stayed in touch, and you were kind enough to join our advisory committee as well. Just lastly, what are you really super excited about from the standpoint of looking to the future?
Yeah, I love it. So yes, it's great to be with you, and I'm excited to have these conversations, because I do think we are at, we can say this probably every few years, but I really feel like we are at a place within our industry where we have a lot of the knowledge, we have a lot of the tools, things are improving so fast in terms of just our technical abilities, as well as, the amount of time and, quite frankly, money it takes to generate some of this information, where I think the next 10 years, the explosion of data that's really going to shape the way, we change how we manage and treat patients is now. We talk about, you'll see in the news and others, you'll see this concept of early cancer detection. I've got a couple kids in high school. The reality is, in their lifetime, they're going to be going to get a blood test like I go and get a cholesterol test to see if they have early signs of cancer, to then do something about it before it becomes a real issue for them. This is the time we're living in. And if you really think about it, it is really exciting.
I couldn't agree with you more. I think that there's so many of us that are really dedicated to this journey. At some point in time, we are all patients, or we have loved ones who are patients. And I have to say, having been in the business for quite some time, it's never been more exciting, I think, from a technology standpoint, and how we're pulling it all together. That's really going to be making a difference. So, but I really, truly want to thank you again for joining us. And I hope everyone enjoyed the questions and answers that we had today.
Well, thank you. And thank you, everyone.
Thank you, Robert.